Right, this was quite an amazing introduction. So it's actually quite a pleasure to be here because in addition to learning many things from Holly, running a lot of work and many arguments with Klein over the year. We led to some papers. That and coffee led to some papers. Even at the very beginning, Noah Magnon, who's here in chemistry, wrote what is really the first paper on sort of landscape theory in which molecules sort of very slowly change the shape and then there's some fast chemical reaction as they change the shape. And that had a very direct and important sort of impact on my thesis work. And so I've been writing up that work. Okay, so this is what happened. In the course of doing experiments on, as Tali said, on the vibristic system, on sensory motor systems in rats, we set out to do some imaging experiments. We set out very innocently to do some imaging experiments. And we wrote a profoundly boring paper because we had all sorts of excess noise. This was with a fellow named Kerry Delaney. And about a year later, we realized, of course, that the excess noise was coming from blood flow, and maybe the blood was interesting because it was noisy. And this sort of led to a foray into trying to understand blood flow in the brain and the architecture of blood flow in the brain. And it just kind of took off, and a bit of a story developed. And I thought it would be sort of a nice thing to give as a general lecture. And I also thought it would be nice to sort of buttress Josh's lecture this morning. I mean, Josh is the height of technology in molecular technology, and I'll try to introduce some sort of optical technology, just to sort of give you ideas of how this, just another leg of this sort of genetics, indicator molecules, and optics revolution is sort of pushing neuroscience and brain science forward. Okay, so the joke is, a long time ago when people gave physics talks, they'd come in with a stack of view graphs, and you look at the stack, and if it was maybe 5 millimeters, they had maybe an hour's worth of material. You were safe. If it was a centimeter, you got nervous. Because the biology talk, they come in with a carousel, and it was quantized. It was between 0 and 80 slides, 2 carousels. It was going to be long. But with a computer, you could basically have something that approaches 1 million slides. And we could get out here and whatever it's Hebrew year 7,000. So I just. Truth in lending, this is our advertising. This is where we are. Okay, and I'll start off. Okay, so I tried to keep it not as much as I'm capable. I tried to keep it not too technical. Okay, so this is actually just about blood flow in the brain and control of blood flow. This, of course, is one of Josh's very famous animals in which the dendrites are lit up. They have a fluorescent protein in them, but we're actually later. It's a completely cold animal. We can see hundreds of microns down, just to give a flavor that you really can have an awake animal, close skull, get away from a lot of problems with the craniotomies. But you see two things. I mean, one is that, of course, you see a number of cells, the dendrites from a number of cells. But you also see all these large objects here, which are red objects, because we've actually stained the plasma in the blood with a dye. And this is the bloodstream. And it's a fraction of the brain. It's a few percent of the brain. And I wanted to describe a little bit about what's happening with this flow. Okay, so first, important slide is who did what. So I'm going to talk about work that sort of spanned a period of about 10 years, but the more recent work I'm going to focus on is doing an excellent Israeli postdoc, Pablo Blinder, another postdoc, Andy Shee, Phil Tsai, who's a graduate student, stayed on as a research scientist, Beth Friedman, who's hopefully here, and Pat Wyden and Jeff Swire. Lasers are dangerous. I'm trying to do the skull. I'll try to be careful about pointing. Okay. And I should also note that over the years we've been funded through the U.S. Israeli National Science Foundation. Okay, so why should you be interested in blood flow? What's special about this? Well, one is that blood is a limited resource. Basically, if you try to think too hard, you'll run out of blood. So there's a need for dynamic allocation of a resource. So in studying how blood is moved around, you could actually learn something about how biology works in resource allocation. The other issue is that they're highly intertwined. So as I mentioned, the distribution of blood impacts on neuronal processing. There's energetic issues. And then there's also sort of practical, clinical issues that come up. Stroke, all sorts of vascular dysfunction or diseases that lead to neurological decline. So understanding something about the vascular system is a step towards understanding sort of basic issues that lead to dementia. The other point is that blood flow and in particular the oxygenation of blood are surrogates of neural activity. So it's impossible, of course, to go to a neuroscience meeting today and not hear about functional magnetic resonance imaging. And this is a very powerful non-invasive tool with which we use to study the brain. But truth be told, and I'll get back to this at the end, the interpretation of the data relies upon rather poorly understood electrical dynamics and neural activity. So I think one really has to understand these fundamentals before one can make interpretations of what's happening in an fMRI image. Okay, so our motivation, in fact, was much more prosaic. As Ty mentioned, that came from physics, and we just got very interested in the network topologies I'll describe and the dynamics of the flow with relationship to that topology. Okay, but then we have to make progress. So what do we use? We're going to use mice and oops, it should say head-fixed anesthetized and awake head-fixed mice and rats. I'll explain why we do it. For the purposes of studying blood flow, a mouse is a truncated rat, okay, in terms of its topology. So this was just luck of the draw that it worked out very well. But a mouse and a rat are not a human. You have a lot more white matter than rodents have. Okay, so there are quite some differences. And the optical, I'm going to introduce a number of optical tools. I mean, we heard this morning from Gideon's talk about two-photon imaging invented by a good friend of mine, Winfried Denk. I'm going to use this to track red blood cells to measure neuronal function. But I'm also going to introduce other optical tools in which you could actually target flow in individual vessels and clamp off vessels at will. And I'm also going to talk about using optics to do automated reconstruction, to actually reconstruct all of anatomy in a sense with using optics as both a means to cut as well as a means to itch. Okay, so there's two principles in this game. Okay, so you set up your laboratory. The first thing you need are animals, and we're going to need access to these animals. And the second thing you need is you got to, right? This is charted, well-charted territory, vascular flow. So you need a way to differentiate yourself from the world and get yourself breathing room. So you need a toy. You need a way. In our case, we need a way to see deep. And our way to see deep is to use the nonlinear properties of optics to their greatest advantage. And I think they're illustrated best in the slide that was done by a fellow named Chris Schaefer in Eric Mazur's lab at Harvard. And what Eric is doing is focusing pulsed light, okay? Light that has an electric field, which is on the order of the energy you need to ionize an electron out of an atom, okay, into the center of a piece of glass. And you see this bright spot. And even though you think of glass as just clear, if the electric field is strong enough, okay, the light interacts. And in this case, we're actually melting the glass at the center. And this is actually a special technique because it does this without heat. Okay, so just to get a little flavor about, again, how you can use some of these new methods to your advantage, this is the greatest problem in doing neural imaging, is you have to make a hole in the skull. Now, it sounds trivial, right? You know, you have all these theories, and at some point, you got to get through the bone, which is terribly scattering, and you got to get down to the brain. And it's not trivial. So, okay, we can use drills, but we also thought about, just as a flavor of how you can use technology to sort of automate physiology and neuroscience and turn it into an industrial process. And on an art form, we could use light to start to vaporize the bone. But then the problem is you don't want to go all the way through the bone, right, and vaporize the tissue. So then, of course, as these ionized atoms come back together, okay, they give off spectrum. Sodium has this famous characteristic line at 590. It's why all the lights are yellow when they're sodium lamps. But if you're in bone or calcium lines, I could use the atomic spectra to my advantage. So this and a couple of other techniques we can pull together. And the bottom line is we could just use light. Cut nice holes in the brain, through the cranium, and look inside. So I'm going to, as I go along, I'm just going to try to describe a number of techniques. So the techniques and the science sort of went hand in hand. Not every newest technique was used with every bit of science, but just to broaden your outlook, to try to think of turning art forms. Physiology suffers because it's an art form. You want to turn it into an industrial process. Okay, so let's get a flavor for what happens. So we've made our hole in the skull. I'm looking down at the vasculature. There are arteries. There are veins. This is a movie by Andy Shee in the lab. The stripes here are just due to the streaks as red blood cells move by. I'm going to be looking at a particular vessel that we call a penetrating artery. We'll learn in a moment. This is what sends blood deep into the brain. And just to give you the flavor that looking at vessels is looking at a dynamic quantity, because we're doing a manipulation here. And the effect of this manipulation is, in fact, it's increasing the size of the vessel. It's making a larger radius. Now, the amount of blood that can go through a vessel is very, very sensitive to changes in radius. It's sensitive because changes in radius affect the viscosity, and it's sensitive because already laminar flow has sort of this radius to the 4th power. So little changes by whatever means, neural control activation, are big changes in flow. Okay, so now, all right, I think Microsoft doesn't like you using their products on a Mac, so it's always a little slow. Okay. So now let's start to get into some physiology, and let's get some idea of how the brain actually rearranges flow. So I haven't said anything special about the architecture yet. I just want to give some flavor of what the dynamics are. So this was an experiment done by a visitor in our lab, Anna DeVore, who received actually her doctorate here with Yossi Yarom. We had a lot of fun doing this. So everybody knows if you go in and you stimulate some part of the body's surface, like the surface of the skin, the surface of the foot, there's a primary representation in sensory cortex. There's a place where the electrical activity is maximized. And this is a forepaw of a rat, and this circle is the place where the electrical activity is maximum, just found with electrodes. And now what we're going to do is we're going to measure the diameter of these blood vessels, similar to what I showed you before. And what you see, in fact, is if you're close to where the center of electrical excitation is, this is the diameter as a function of time. Diameter is a function of time. So very close, all of a sudden the vessel gets bigger. It's a large effect. It's like 15%, almost 50% increase in flow. But if we go rather far away, you see that there's actually a net constriction of the vessels. So the surface is somehow acting to take blood as far away from where activity is and cut its flow, and take blood close to where the activity is and increase the flow. And the most efficient way to do this is to have a network, is to have a sort of a system of all sorts of interconnected loops. Now, whether it's a Manhattan grid that's very regular, or it's sort of like the streets of Jerusalem, which just from walking around seem rather irregular, nonetheless, they're highly interconnected. So let's see what the system tells us. Okay, so as you've seen, the surface arterioles are dynamic. They could dilate or constrict to redistribute blood flow. And our hypothesis is that this results from a highly interconnected surface. Okay, so we look. It's very simple. So we take many animals. We do a number of techniques, flatten the skull, fill it with dye, etc., etc. So this represents all of the blood vessels in one hemisphere, okay, of the rat. And the greens are the outlines of all the surface arteries. I'm ignoring veins for now. And if you steer back, the critical thing is that we colored all the vessels that form closed loops in black. So the major architecture of the major topology, you would say, of the surface is a topology of closed loops. It's not a topology of trees where blood goes and branches and branches and branches. So what this means is that any spot on the brain gets fed by blood going this way, or that way. There's all these different pathways. And we can analyze this, and I'm just going to show one sort of not so much analysis, as sort of categorization of what's happening. So we can just take this backbone, we could look at it, and one thing, the simplest thing to do is to say, well, if it was a tree, then the number of vertices, which are places where vessels meet, they always meet at threes, okay? And I could look at how the number of edges, which are the branches between vertices, scales as the number of vertices. So if it's a tree, they scale 1 to 1, okay? If it's a lattice, like a honeycomb lattice, it basically scales 4 to 3. So to sort of zero approximation, okay, just if you abstract it, the surface looks like a honeycomb. No, yeah?
Is it unique to the brain, or is it all over the place in the body?
No, this is actually fairly unique. So I could discuss a little bit other areas. I mean, there are, this notion of closedness is seen everywhere. I mean, you even see it in Harvey's picture of the vasculature going down the arm, and you can see that the arteries are actually closed. But there's no other place where you have this greatest density on top. And as I'll get to, this is only one of 3 architectures. So that's what made the brain interesting. As you'll see, there's actually 3 different angio-architectures that serve 3 different roles. Okay, so it's not a perfect honeycomb. In fact, if you walk around all the loops, the fraction that are 6s, it doesn't peak at 6. It peaks at 4. It's imperfect. There's a rule you have to write down. But this is the flavor. You can just think of, I have a honeycomb, and it's highly interconnected. Okay, so the first question is, does this really lead to robust circulation of blood? And you have to ask how you would test it. And a nice way to test this is to say, well, if this is true, what I could do is this. I could take my picture of the surface. Again, there's a lot of veins, but I could just factor out the veins. And I could plot only the arteries. These are all the loops I talked about. And I can say, well, wait, I'm going to block one of the loops. Now, if it was just a tree structure, then everything downstream goes dry. But if in fact there's a loop structure, then what I should see is one of the downstream vessels flip its direction of flow. And I should see flow roughly restored to normal levels due to this flip. So let's do this experiment. Okay, so we need two tools, as always. Right now I need to follow the blood flow. I need to actually be able to quantify the blood flow. Okay, so we could do this in very large vessels, but I think to illustrate the point, I actually just wanted to show how you actually follow blood in the smallest vessels, capillaries, just because it provides the most graphic explanation. So we can image deep into cortex. This is one capillary, small vessel. You see it's very small. It's about 5 microns. In the rat, it goes down to maybe 2 microns in the mouse. And then what I could do is I could just put a little box around this region and take a series of snapshots. Like one snapshot, in this case, it's one every 15 milliseconds. What you see is sort of white regions. That's where the dye is in the blood plasma, and dark regions. That's where the red blood cells are that are occluding the plasma. Now, if the red blood cells move, then expect the dark spots to move in time. And in fact, what you see are these dark spots here moving to the right. So this constitutes a measurement of speed. The distance between them corresponds to sort of a measurement of the density, right? And the number that passed me by constitute a measurement of the flux. So I could automate. Now, in practice, what we do is we actually just scan back and forth with our laser beam, and things go from bright, where there's no red blood cell, to dark, where there's a red blood cell, and back again. And as I keep on scanning sequentially in time, I build up what's called a space-time plot, which are just bands, and the slope here, 1 over the slope is a measure of the speed. Okay, and this could be automated, so you could just be sitting there sort of watching this in real time, analyzing it, at least now, in real time.
Why are they clustered?
Why are what clustered?
The red blood cells.
I mean, why are they next to each other?
No, why are they clustered? Why are they not spread around?
But they are spread. This is one. This is one red blood cell.
What is in between?
This is
okay,
just
one. Okay.
this is blood plasma, just proteins, platelets that are not resolved. Yeah. Okay. Okay, everybody. Okay, so that's tool number 1. Then we need a second tool. We need a way to shut the vessel. Now these are tiny. These are vessels that are 20 microns. So going in with thread and that's beyond me. So again, we appeal to using light to our advantage. There are molecules in the literature called photosensitizers. That's a fancy way of saying if you shine light on these molecules, they make free radicals that live for a couple of hundred microseconds, and they'll react with anything in the vicinity. And you could use that to your advantage because we can inject these photosensitizers into the bloodstream. They actually live in the bloodstream for a few minutes. And then I could shine light on, and I could shine light which is just at the threshold for doing damage. Now these vessels are on the surface. So if in fact the light is just at threshold, at the focus of the lens, then as the light starts to splay out below the lens, I'm below threshold, and I don't cause damage. So there's obviously a number of control experiments behind this. But you can see the effect here. So this is, I'm sure there's some traffic intersection in Israel that looks like this. This is blood going in. It bifurcates once. It rapidly bifurcates a second time. This is just a control experiment. I shine green light, which activates this molecule, rose-bengal. Nothing happens. But if I add rose-bengal and shine light, I start to form a clot. And this is very slow exposure. And after a while, I completely block this vessel. It's bright over here because I'm just backing up with blood plasma. And already you could kind of see already the answer because the direction of flow in the vessels below changes. Okay, so this is N of 1, as they say, but I could do this in many cases, and I'm just going to illustrate an example up here. Okay, I'll break down the... Wow, it's 1 weak laser. Okay. So this one won't burn a hole in your skull high. Okay, so this is one of the loops that I talked about. What we did is we went ahead and we made a block at a point, and you can see that flow, which initially in this direction reversed itself. The magnitude of the reversal is on the same order. Okay, you get downstream changes in flow also. But the bottom line is this. I mean, you need to knock down flow by something by almost 80% until you have a physiological effect in the short term. And if we block flow and we look at what happens in the downstream vessels, is that the flow drops on average to about half its value. It always reverses, of course, in one of the vessels. And if we look further downstream, in fact, we're essentially at the original levels, okay? That essentially at 1. That there's, on average, there's no effect. Okay, so the bottom line is that the same surface vasculature that lets you redistribute blood to give to areas of need during electrical activation, of course, you can check your ideas. It's due to a highly interconnected lattice, and the same lattice gives you a certain protection against the block. Okay, so it is of interest that clinicians, as best one can tell from the literature and from talking, never see sort of microstrokes on the very surface of the brain. Okay, so we said that blood flow on the surface is insensitive to single occlusions, and now let's get to this structure called penetrating arterioles. I mean, this is the business end. These are the vessels that take blood from the surface, right? They're just circulating the surface, haven't fed the brain yet. Dive into the brain, and let's see if this surface vasculature actually keeps the vessels that diving into the brain insensitive, okay, to an occlusion. All right, so now we need another trip. We need to learn something more about the architecture of the brain. So we have to start to study the vasculature of the brain, which means this led to sort of a large scale project in the lab, which in the days of omics, we call the angiotome, right? Something for your funding officers. But again, we need a way to do large scale anatomy to really look at a lot of vessels. And again, I'm just going to take an aside. Our lab and other lab, Bruno Weber in Europe, have sort of gone back and re-looked at vasculature, and all sorts of things that were sort of in the early literature seem not to be true. So you really need to look at many, many samples. You need to reconstruct large areas of tissue, because if you just look at a little piece, you know, there's so much inhomogeneity and so much variation, you could rapidly come to the wrong conclusion about where vascular density is high and low relative to neurons. Okay? So our technique for doing this, again, makes use of the same light, okay, that's capable of cutting and vaporizing tissue. And the idea is that we image until we run out of signal to noise using two-photon microscopy, run out of signal to noise, come with our amplified laser light, vaporize off the surface, and then image some more, run out of signal to noise, come with our amplified laser light, and image some more, and just keep on going. So in practice, you're taking the tissue, you're sucking out all the signal to noise from all the fluorophores, and at the end of the day, you know, essentially you're left with dust. Okay, but you can take all that data, and then you can reconstruct it. So this is raw data. This raw data then goes through a fair amount of analysis, and it's turned into numbers. So numbers like where the center of cell soma are, which I've not shown you in this picture just for simplicity, and vectors describing all the vessels. So based on surface vessels, this is a small piece of the brain. This is 1 millimeter. The big piece I showed you before was like 20 millimeters. But we can see vessels that come from the surface and drop down. And these are the penetrating arterioles. They take blood from the surface, and they drop down. And as they drop, they give off branches. Sort of a feature about neurons to worry about at the moment is that you have these golden bands, and these correspond to the location of cortical columns. So this is the vibrissa area of the rat, which is bird studies, and it's sort of well known for the representation of each hair having a very tight representation in neurons. Okay, so let's look at these vessels. So if we go back and now look at the tops of these vessels, so before they dive, just as they're starting out, again, if the surface vasculature just gave rise to a tree structure and lots of vessels left that tree, we've lost all our immunity for defects. But on average, what happens is this, from sort of studying these maps of how the surface of how the penetrating vessels start. This is a surface vessel. It has a little stub, and it dives. And this represents about 89% of the vessels. And these tree structures are very infrequent. Okay, now why this falls off like parallel, I thought I'd ask all the theorists here to explain this to me. But I think from the biological point of view, the critical thing is that each vessel that dives down doesn't branch. It just leaves and dives. And that makes it rather immune to single-point defects. Okay, so how do we do? So we do a very similar experiment to what I showed you before, where we're going to block one surface vessel, except now, instead of worrying about what's happening in other surface vessels, we're worried about what's happening in the business end. What's happening to the flow in these penetrating vessels that are supplying blood to all the underlying neurons. And the data is shown here. So we want to attend first to the green dots, okay? And what's happening is that for some values of initial flux, blocking a surface vessel has absolutely no effect on how the brain is fed. So you really see the safety factor coming in. And for other vessels that were initially very large with a lot of flux, there's a bit of a decrease, but that decrease is too small to be of physiological damage. And the black dots here are just looking at penetrating vessels, which are more than one branch away, which is from the data before you would expect there would be no effect at all. Okay, so the bottom line is from this is that there's a surface vasculature. The surface vasculature redistributes blood, and the surface vasculature also endows the vessels that feed the brain with an immunity from single defects. Okay. So now I want to go back and I want to focus on these penetrating arterioles. They seem kind of funny, right? Because they're diving straight down. So if the surface forms a two-dimensional grid, the penetrating vessels seem like they're very one-dimensional. And that gives you some clues. Maybe this is where, in fact, you're controlling the blood flow, because these vessels are also coated with smooth muscle. But maybe also that these are sort of bottlenecks to flow, that blocking one of these vessels could actually be rather severely damaging. Okay, so I'll go back and look at to remind everybody what these vessels look like. This is a slice, a virtual slice out of this reconstructed vasculature. The top is the surface, and you see these vessels coming straight down, and off of these vessels are these very fine microvessels that feed the brain. So I'm going to look at what happens, what the consequences are of blocking one of these. Now, the thing by looking at this is it's not obvious per se what the answer is going to be, because here's a penetrating vessel, here's another penetrating arteriole. So it could be that if this one can no longer supply blood, and the fine vasculature coming off this other vessel might provide collateral flow. So the resistance in these things is very sensitive to the radius, and they're small, so they just might be too high impedance. On the other hand, one vessel may not really matter. You make a strong collateral flow. So I think you sort of have to do the experiment here. So let's see what happens. So we go back, and again, we could map the surface. We could find out where all the penetrating vessels are. We appeal to our optical tools to make a block where we want. This is important. We can measure all the flow parameters before we make a block, and all the parameters after we make a block, and then we could use a two-photon microscopy then just to confirm that flow stops in this penetrating vessel. So it's a controlled system, and this is what you get. So before we blocked the surface vessel, you lost a little bit of flow one vessel away. But here, you have a completely catastrophic effect. We're going to block the penetrating vessel here. And then what we did is just walked sort of through layers 2 and 3, sort of through the upper sort of 100 microns below the surface to 300 microns below the surface. And we measured the speed of flow in as many vessels as we could. And then we measured the same speed after we blocked a single point. Now, there's a lot of scatter, and the scatter represents biological variability. But the point is that you've lost your flow, and you don't recover your flow until you're out. So you're out to a radius of close to 300 microns, or diameter of something like 500 to 600 micron. So blocking one vessel has basically killed an entire cylinder of cortex. So this shows, I think, rather dramatically that these penetrating vessels are a bottleneck in the supply of blood. Very, very different than the surface, maybe not inconsistent with the low dimensionality. And one question, so there's a medical question now that comes up, is what's the chronic consequence of this? Do you actually redistribute flow over time? So there's two ways to look at this. One is from the point of physiology. So one of the things that happens is when you lose flow to vessels, or when neurons start to die, is their last gasp in the world is what's called spreading depression. Spreading depression is where you get a wave of electrical activity, a wave of depolarization. And you can see this with a surface electrode, or as the cells depolarize, calcium rushes in. So we use in this case, we use a calcium indicator. These are molecules that change the fluorescent properties upon the binding of intracellular calcium. It's familiar to a number of you. You make an injection, uses a molecule designed by Roger Chen and a technique invented by Arthur Connereth to have all the cells in a region filled with this dye. But in this case, it's simply a measure of activity. And what you see after you block this vessel is one of these waves, slow waves, the last gasp of activity, spreading depression, move across cortex. Okay, and the consequence of this last gasp is shown here. So this is a surface vessel. This is now a projection from the side. We've imaged into a cube. I'm looking at the top. I'm looking at the side. This is a penetrating vessel. I come in and make a block. I've lost flow in this vessel. I come back a few days later, actually 7 days later, and I have an animal. This is a transgenic animal in which the microglia, so the microglia are sort of like, you can think of the macrophages of the brain. They contain chemokine receptors that go after dying cells, and they completely infiltrate this dying region. And you literally see a column due to the death of one vessel. Okay. So the consequence of this, we can now come up with a picture, right, from all these experiments of different sort of cortical, different sort of cortical topologies. So this is, if I didn't show data, I'd get very beat up for making such a cartoon. But let me give you a flavor for all these scales. So we talked about a surface network, right, which redistributes blood, which the zero approximation is kind of a honeycomb. So that's this backbone. Coming off of this honeycomb structure are these penetrating vessels that dike down. So these are these green dots. They come off simply as stubs, and the stubs give you a certain protection. Okay? There's a region of neural activation, if you were to touch the skin or touch your whisker, and that's about the size of this golden dot, okay, on this scale. So it actually encompasses multiple loops on the surface network. And then there's a region, a cylinder that dies, okay, basically a cylinder that's fed by a single penetrating vessel, and that's given by the silver dot on top. So it's small compared to an activating region, okay, and we're doing behavior in the lab to see if, in fact, one of these doesn't cause a behavioral defect, but a few will. So this is sort of a scale for how things are fed in cortex. Okay, now, the question is how new is this idea of loops, right, loops on the surface? And in fact, it's an old idea. It's a 350-year-old idea because it goes back to the circle of Willis. So if you were to look at as blood comes up through the common carotid and through the vertebral carotid is here, the common carotids are here, they meet. And the two carotids, the vertebral carotids, form a loop. Okay? This is the so-called circle of Willis. So at the very base of the brain, okay, before the great cerebral arteries come out that feed the cortical surface, you have already one piece of redundancy. When they feed the cortical surface, you have another level of redundancy. But once they actually start to feed the neurons themselves, that's the end of the redundancy in the feed system. Okay, so this sort of posed an interesting, we like I said, many unexpected things were found. And sort of before I get back to straight physiology, this sort of posed an interesting medical issue because it's true that people have strokes, I mean, in the sense of presenting some severe neurological defects. But then there's a more subtle kind of strokes that over time, there were a number of studies done. These are the Rotterdam studies where patients, I should say, individuals were followed, older individuals, as they write in the paper, year after year after year, and they will take an MRI scan, and you start to pick up water-rich regions in the brain, okay? These are known as UBOs in the fMRI world, unidentified bright objects, as I've learned, although people usually don't write that in their papers. And so it looks like you're getting these little dead regions in cortex, and we thought that perhaps they just correspond to single blockages, okay, of penetrating arterioles. So we started to move in that direction just to see if there's some connection. And I showed you that blockage of a single penetrating arteriole leads to spreading depression, eventually cell death and a cyst. So one question is if there's a synergistic effect, because one penetrating vessel had a small effect on the scale of neural activation. So what happens if you have a number of these defects together? So again, just sort of rolling this technology to ask sort of a more medically bent question, we can make a number of blockages. This is work by Andy Shih. And the point is this. If you have an isolated defect, this dotted line here, it's small. It's like I showed you before, 400, 500 microns. But if you have a number of penetrating, if you block a number of penetrating vessels that are reasonably close to each other, even though there's flow, as I'll show you in a minute, in the region in between, over time, they all coalesce. And you get a very, very big defect. And this is sort of a hypothesis that a lot of dementia sets in, and sets in very quickly. All you need is a few vessels that die, and the space in between all of a sudden will fill in. So this is bad, okay? But there's good news, okay? So once you have the ability to control things, and just sort of suggesting how one could sort of use sort of these modern sort of neurological tools, imaging tools, then you have an ability to test things, okay? So again, this is work by Andy Shih. And there's a lot of work, there's a lot of neuropharmacology in the stroke world to worry about preservation of tissue after strokes. But these are severe strokes where large fractions of a hemisphere or basal ganglia died. But we had a very focal stroke. And what we did was we repeated the experiment I showed you before. These are places where we made blockages. You come back after, in this case, 2 days. You see the vessels in between have stopped flowing. I've shown these in red, vessels that have stopped flowing. This is basically the previous data I showed you, but now I'm indicating why you get a cyst. You just have a total breakdown of flow. But if you do the simplest of pharmacological manipulations, which is to put in a rather strong blocker of excitotoxicity, okay, this is a molecule that actually binds very strongly to the glutamate receptor, to the NMDA receptor. What you find is if you do exactly the same experiment, you come back days later, there's no breakdown in flow. Okay, so we're sort of in the middle of this, but just to give you some flavor of how by being able to manipulate one vessel, or it could be one neuron, you could actually go in and sort of settle some very nice neuropharmacological issues. Okay, so
Can you
yeah.
repeat again what was the manipulation?
Okay, manipulation was adding this molecule MK801. This is a street drug. It's angel dust, and it blocks it's a strong blocker of the NMDA type glutamate receptor. So you sort of
But the entire tissue? Or
walking.
But where do you put it?
We actually put, in this case, it's given systemically. Okay, it's just very simple. This was given systemically. It wasn't just a local patch. Part of it is that we have a window over this, and we don't. So the sort of the backstory to this is just making a window itself will cause the blood vessels to start to rearrange. Okay, that the craniotomy itself has problems. So you really want to do things where you could give things systemically and avoid craniotomies as much as possible.
The mechanism or extremely?
No, okay, so I said preliminary evidence. That's sort of motivation. Okay, trying to give some motivation for how. So I could guess, but I don't know the detailed mechanism. That's why I wrote preliminary. Okay, just try to get you guys thinking of how, if you have a new way of doing things and you sort of follow what's interesting, things happen. Okay, so I talked about, this is our hypothesis, that these cortical microinfarctions, which are seen clinically in vascular dementia, are a consequence, perhaps, of knocking vessels down one by one. And I think this is maybe a medical front, a clinical front to think about. But there's another issue, a much more basic issue, is that we lost flow, right, in sort of a column of vessels. And I raised the point as to how does this happen, okay? Does this happen because the vessels form a module, that a penetrating vessel comes in, forms a capillary bed that stays local to itself, and then drains out through penetrating veins? Or does it actually just form the cylinder because it's just too high a resistance, too high an impedance for neighboring vessels to push blood in? So the only way to answer this question is to put some tracer in that will just follow along the individual vessels, or to actually do a large-scale reconstruction of the vasculature. Okay, so we took a more brute force approach. Okay? So let me give you a flavor for what's going on. So this was all the vasculature imaged, like I said, with this cutting method, and we're reconstructing it. I'm sorry for the low resolution, but in reconstructing it, we turn the vasculature into vectors. By turning them into vectors now, we can do things like compute flow. It's a little dark. This is the surface vasculature I talked about before. Just give you a little bit of a flavor. These are cortical columns. These are the barrels that are outlined. Other things pop up. Turns out most of the penetrating vessels are right around the edges of the barrel, so the way you build a lab, you put the utilities around the edge, and then I can start to fill in the cells. But again, now if you do this quantitatively, the cells actually correspond to a location in space, a vector and a sphere. Okay, and I'll just stop here. Okay, try to stop here. Okay, so we have this very fine vasculature, and once I have this very fine vasculature, I can start to do math on this very fine vasculature to answer the question: are these things modules or are these things continuum? So I should add, this is actually the work of Paul Linder and Bill Sy. So to look at the entire vasculature together looks like a plate of spaghetti or udon noodles or whatever your favorite pastime is. So the one question to ask is, I know where the cortical columns are, and I'm just going to look at the vessels that sit within this one layer, this middle layer of cortical columns called layer 4, and let me see if these are interconnected, just to give you some flavor of the problem. And in fact, if you start in a given vessel, you'll work your way all the way through all the vasculature. So there are no modules. In fact, there's no relationship at all between the microvasculature and the placement of the cortical columns. I think this is very important because it bears on fMRI as well, where there's sort of this notion that blood vessels actually follow, okay, a columnar representation of neurons. So at least as far as all the somatosensory cortex goes, in mouse, the relation is zero relation, okay? And the other issue is that now we can follow around these loops, and we can see if they form a closed loop structure a little bit like the surface. And the loops are larger, they're coming in sort of double the length, they're peaking at sort of 8 edges per loop, but we also form this loop structure towards the bottom. So the microvasculature also has this loop structure, and we could again ask the same kind of question. If it's a loop, what the consequences of a block? Do the loops actually keep you from blocking? Now, this is actually an important point because at any given time in your brain, about 10% of these microvessels don't have blood flowing in them, right? They're just stuck. Okay, they're stalled. Okay, and you can see this very nicely. I didn't show any pictures. You can see this very microvessels, in fact, okay, that the flow should recover in further downstream. That blocking one of these loops should not stop blood flow downstream. So again, now we're trying to block a vessel deep in the brain, and we can't use our simple linear technique of shining light on the surface at threshold, because if we shine light and try to have a focus, first of all, we'll never achieve a focus at depth. And the second thing is if we tried, we would just block everything on the way down. So we use our nonlinear optics. We focus deep. There's no effect due to the light until we get to the focus. At the focus, then we make a little nick in the vessel. One consequence is that nick leads to a shock wave. You can see this very nicely. The shock wave actually causes serum or plasma inside the blood vessel to come out, but the blood cells keep going. It just causes the walls to rupture a little bit. It's a way of getting molecules into the brain without breaching the blood-brain barrier, if you will. But with a little more judicious control, we could actually cause the blood vessel to completely block. So you have some control. And the experiment is very, very straightforward. We make a block, and we see what happens downstream. And we look at the change in velocity downstream from a block. These are 5-micron vessels. Now, one vessel downstream is almost 100% block. But if I go 2 vessels, 3 vessels, 4 vessels downstream, okay, the change in velocity is rather slight. So again, these large vessels, not quite one vessel downstream, but further downstream, allow flow to be maintained. So again, these small vessels are probably functioning on some micro level, actually, to maintain flow. Okay, and the other point is that if you kill one of these small vessels and let the animal grow up, this picture is impossibly dark up here, so I'm just going to say in words, killing one of these small vessels leads to very little damage. I could look for dead cells with a molecule called propydium iodide. And you see 2 or 3 cells that are dying right next to the vessel. And this is very, very different than in control data, where you block penetrating vessel and you have, again, this column of death. So if I did this correct, I could summarize and I could say this. Blockage of even one vessel, okay, leads to some cell death. Right? A few neurons will die, but it won't lead to a cyst. So at some level, every blood vessel in your brain counts, right? Some give you a more severe consequence than others, but everyone counts. And I tried to summarize this as follows. This was a summary of data taken over a number of years, and the sheet put together. If you block a penetrating vessel, like I showed you, or even a penetrating venule, the return supply, there's a certain number to how much flux goes through these vessels, but you get a cyst. You get a region which will never recover. If you block multiple vessels, you get an even larger region. It grows in a very nonlinear way. And if you go to some of these second-order branches that are just coming right off the vessel, you still get a cyst. But if you look at any of the vessels that form cyclic networks, either on the surface or in the microvasculature, okay, you don't get a dead region. So it makes kind of sense. The one-dimensional objects lead to neuronal death. The objects that form loops act in a protective way. All right, so this is kind of the summary, is that there's the surface vessels are the two-dimensional distribution network. The subsurface vasculature looks like a three-dimensional grid. They have a certain amount of redundancy. This redundancy, at least on the surface, functions to redistribute blood as needed. It also has this sort of secondary, you might say, medical benefit or clinical benefit. We'll see. The penetrating vessels are one-dimensional conduits that take it from the surface all the way down. Okay, and these are bottlenecks to flow, and these, of course, are extremely sensitive to any kind of damage. Okay, so the last point is just, so we're sort of finishing up this full sort of three-dimensional reconstruction of the entire vasculature of mouse cortex. But we want to start thinking about the control, about neuronal control of blood flow. And in particular, the penetrating vessels have smooth muscle about halfway down. Okay, so they really seem like the place to focus on. And I want to give a little bit of a flavor for why the question is important. And we don't have any answers yet to how the flow is controlled. But I just want to set up the problem, that it's a not trivial problem, and give a little bit of a flavor, again, of how one could use sort of modern neurological tools to attack the control of blood. The same tools you'd use to study neural circuits. So this again, is an experiment done with Anna DeVore visiting our laboratory. And what we wanted to do here is look at blood flow in response to two different means of activating cortex. Now, I mean, everybody knows if you poke your right foot, it's the left side of the brain that gets activated. But, if you poke your left foot, it's the right side of the brain that gets activated. But it's also the right side of the brain, because there's two ways to get information in. It could come, through the dorsal column and goes over to the contralateral side, but then there's a second pathway, right through the callosum, that takes it to the same side, to the ipsilateral side. So you're activating neurons in different layers. So that constitutes a contrast mechanism for neural activation. And let's see how blood flow tracks, okay? There's two different pathways in neural activation. So this is a very simple experiment. We're stimulating the forepaw of the animal, okay? So we're stimulating here on the right side. We're measuring on the left side. We're recording with an extracellular electrode. None of these details really matter. You get this large burst of activity. Very, very, very naive experiment. And if you go in, we find the center of electrical activity. These are these big red spikes. This is the surface potential. And what you find is near the center, the predominant response, the dominant response, is an increase in the size of the vessel. So this all makes sense, right? Electrical activity goes up, blood flow goes up. Vessels get bigger, and blood flow goes up. Okay, let's go to the other side of the brain, the ipsilateral side. Again, we stimulate, just as we did before. The response on the ipsilateral side is somewhat delayed. Look at this time, it's much longer than on the contralateral side. Okay, response is a little weaker, but there's a clear electrical depolarization. And if you use other techniques like the voltage-sensitive dye technique or increases in metabolism, we see an increase. So what happens to the blood flow?
Goes down.
Because otherwise I wouldn't have showed the slide. Okay, you might have been a referee. So it's true. The vessels all constrict. The blood flow goes down. So this is a one-off sort of proof of principle experiment that there's not a one-to-one connection between an increase in electrical activity and an increase in blood flow. And if you were doing an fMRI experiment, this would turn into what's called negative bolt, which I don't know what that means, scare or something. It needs a new acronym, okay? And the question is, how does this arise? Let's try to think through let's try to set up what would be sort of a research program and think through the logic. So there's not a direct connection, okay, between neuronal firing and the output of that firing exciting the muscles. They're intermediaries, okay? And those intermediaries are neurotransmitters. And there's two flavors of actions by which neurons will actually cause the smooth muscle, okay? So this is like a sensory motor system. So neurons are putting out small molecules, transmitter molecules. If you have to, think of olfaction. And then you could look at the motor output as a smooth muscle which is surrounding the blood vessels. So inhibitory cells will make direct connections on the smooth muscle. So some inhibitory cells, okay, so-called baronaric or somatostatin cells, will put out, in addition to GABA, okay, in addition to small terminals, they have dense core terminals. They put out peptides. And one of these peptides is somatostatin. Somatostatin will cause the vessels to constrict. Okay. Other inhibitory cells, okay, such as these vasointestinal peptide cells, will put out a neuropeptide, and they'll cause the vessels to dilate. So you have two different populations of cells, and they're competing. One's trying to cause the muscle to get bigger, and one's trying to causing it to get smaller. Astrocytes. So excitatory cells don't affect the vessels directly. They work through a middleman. Okay, and the middleman is the astrocyte. So glutamate, the excitatory transmitter, will leak out of the vessels, leak out of the synaptic cleft, and it'll gradually activate astrocytes. And astrocytes put out breakdown products of this internal molecule called arachidonic acid. The names aren't important, but they could either put out PG, prostaglandin 2, or they could put out a second molecule, which is abbreviated 20-het. This molecule, 20-het, leads to relaxation of the muscle and leads to constriction of the muscle, leads to vasoconstriction, and PEG leads to vasodilation. Prostaglandins leads to vasodilation. So again, there's a competition between the two. So the game is actually to understand, okay, what the pattern of electrical activation is. And in principle, that pattern should tell you whether you're going to get net dilation or net constriction. So this is a hypothesis based on the previous experiment with Anna and a number of other experiments published over the last few years. So I'm just going to spend the last few slides. So this is a computational group, and how are we going to turn the study of vascular flow into a computational problem? Okay, so the first thing is you write down an equation, right? And what does vascular output depend on? It depends on the smooth muscle tension, so we need a way to measure smooth muscle tension. And it depends on vascular architecture, which is a project which is going on. And what does smooth muscle tension depend on? Well, it depends on the activation of these different types of inhibitory cells, each putting out a molecule which can cause things to constrict or dilate, and it depends on the activity of excitatory cells through the activation of astrocytes. So we need to measure all of these pieces. And just in the last two slides, I'll give you some flavor of how you could do this. So this is very much neuroscience. So these are animals that come from Michael Kotelkov at Cornell, which in their smooth muscle have a molecule that responds to calcium. Okay, so this is a natural buffering molecule, and that's been tagged with two fluorescent proteins. And if the fluorescent proteins get close, they'll have a dipole-dipole interaction, and their fluorescence lifetime will shift, and if they're far away, they won't interact. And that's a contrast mechanism to understand calcium in the cell. And of course, we can measure the diameter, we can measure or flow in the vessel. So just in this one sort of example, sort of proof of principle example, we could be looking at the diameter of the vessel as a surrogate of muscle tension, and we could be looking at calcium. And of course, not surprisingly, as calcium goes up, the vessel gets small. And you can just see calcium signal go up, and you can see the vessel get small. So this is sort of one piece. You see the diameter of the vessel go down. So this is sort of one way to sort of use these new tools, use genetics, use indicators, and use optics to answer this case a neurovaso coupling question. The other flavor is that one by one, you'd like to activate these different constituents that could cause vessel constriction or vessel expansion. So you'd like to go through the list of different neurons, which means you either have friends or animals that other people have made, or maybe you make them yourself to label these different subtypes. And then you want to put in a molecule that could cause these neurons either to be quiet or to be active. So in this case, we use this molecule channelrhodopsin that came from Bamberg's lab, and it was really put to tremendous use in neuroscience by Carl Diesterhoff. So we're exciting now astrocytes, and what happens is every time we cause an astrocyte to become depolarized, you see a jump in the speed of blood. So it's just sort of it's just sort of a proof of principle here, just to give you some idea of the logic that you could use to sort of decipher how neurons control blood flow. So we'll see what happens, okay? So I started out just by, what started out as sort of an accident, right? Extra noise just sort of intrigued us. And what I hope I've convinced you of is that there's sort of multiple different topologies in the vasculature, and these give you some idea of different functions for different types of vessels. And then there's sort of an interesting, underlying problems, some of which relate to disease, and some of which, I think the ones that we're going to focus on, really relate to how these neurons actually control how their neighboring vessels feed them. Okay, thank you.